in Algeria with a doctorate in pharmacy, followed by a master's degree from the University of Montpellier in France. Her research into the hearing sciences, however, began during her PhD studies also at the University of Montpellier, where she focused on unraveling the role of DNA damage in cisplatin-induced ototoxicity. Now an instructor in the Department of Otolaryngology in the Department of Otolaryngology and Head and Neck Surgery at Stanford University, we honor her today for the work she completed there as a postdoctoral fellow in Dr. Stefan Heller's lab into the genomic and transcriptomic characterization of avian hair cell apoptosis and regeneration, work that has helped to identify the triggers of underlying hair cell regeneration in avian cochlea and may ultimately inform work into hair cell regeneration in mammals. Please join me in welcoming this year's Bellucci Trainee awardee, Dr. Nesrine Benkafadar.
Yeah, thank you so much for the introduction. And before I start, I would like to thank the organizers for giving me this great opportunity to be here today and to present my work. So today I will talk about the role of proteolytic activities in controlling avian auditory hair cell regeneration. So as we all know, hearing loss is the third common chronic disease in the world. So hearing loss is caused by has several causes. So we know that it is used by age-related aging, also genetic predisposition, but also due to the loud noises and some ototoxic medication. So the consequence is so it results in the loss of the sensory cells of the inner ear. So in mammals, we know that this is it leads to irreversible hearing loss. So in mammals, there is no turnover and no regeneration. And here we are talking about the cochlea and not the vestibule. And the consequences are severe. Sorry, I can manage to do it. Yeah, it doesn't work anymore. Okay. Yeah, so the consequences on health are severe. So we are talking about social isolation, about problem in communication, but also an increase in stress, in anxiety, and also the early onset of dementia. Okay. Yeah, and in contrast to mammals, we know that the non-mammalian vertebrates, they display robust regenerative potential when it comes to hair cell regeneration. So the non-mammals, they have this amazing ability to restore damaged or lost hair cells, and also they recover a fully function of their auditory and vestibular function. And currently, the avian cochlea is the primary vertebrate model to investigate auditory hair cell regeneration. So the avian auditory sensory organ is named basilar papilla, and the basilar papilla is located in the cochlear duct. The basilar papilla is the equivalent of the mammalian organ of Corti. So if we dissect the basilar papilla and we make a transversal section, so we will see hair cells that are interdigitated by supporting cells. So avians have two types of hair cells. They have the tall hair cells that are located in the medial noocyte and the short hair cells located in the lateral abnoocyte. So we know that in birds, auditory hair cells are regenerated regardless of the source of trauma. So as you can see here in this figure, so we can observe the formation of new hair cells, and this is 10 days after an acoustic overstimulation. So also supporting cells are considered to be facultative stem cells because they are mitotically quiescent unless activated by dying hair cells. What we know is that when a hair cell is dying, supporting cells are activated, and then hair cells will get generated via two different mechanisms. We have a direct transdifferentiation that happens mainly in the abnoocyte of the basilar papilla, and a mitotic hair cell regeneration that happens in the noocyte of the basilar papilla. However, the exact mechanism by which regeneration of the sensory hair cells occurs is still unknown. So our general hypothesis is that when an ototoxic drug is administered, for example, so the hair cells will receive a signal and then we send some signalings. And then apoptosis will start and continue until the hair cell die because they are too fragile. So in parallel, the supporting cell will receive the signal and then commence a regenerative process. So the epithelium will deal with the parting of hair cells and then supporting cells re-enter the cell cycle and ultimately divide asymmetrically and give rise to a new hair cell. So the objective of this work is to understand how the supporting cell react to hair cell damage and also what triggers the onset of cell cycle entry in supporting cells. So, and to answer to these questions, the first step was to establish an in vivo model and here our model consists in a unique injection of sizomycin into the semicircular canal of P7 chickens. So sizomycin is an aminoglycoside that is highly pure and highly ototoxic. So using this technique, we are able to get a rapid and complete loss of the hair cells within 24 hours. We also observe an S-phase entry an S-phase entry in the supporting cell between 36 and 72 hours with a peak of EDU incorporation in the supporting cells at 48 hours post-sizomycin treatment. We also observe new hair cells, as you can see here, labeled with myosin 7A and SOX2, about after 4 to 5 days post-sizomycin treatment. So based on our characterization of the time course of hair cell damage and also supporting cell regenerative processes, we decided to collect single cell RNA sequencing at different time points. So here today, I will talk about the 12, 16, and 20-hour time point as an early time points where we know that hair cells are undergoing an apoptotic process. And here the aim is really to understand or to investigate the early supporting cell transcriptomic changes. And we decided to add another latest time point that corresponds here to the 38-hour post-sizomycin treatment when we know that supporting cells are undergoing already in S-phase entry. And based on this, we have a transcriptomically isolated supporting cells. So analysis of the single cell RNA-seq revealed 4 clusters, as you can see here in this Disney plot. And in the bar plot, we have the distribution of cells by time point and by state. So what we found is that S1 and S2 contained mainly cells from the controls. So as you can see here in green, and we define these 2 states as control baseline supporting cells. Then when we looked at S4, cluster S4, we found that this cluster contained mainly cells from the latest time point, and especially from the 38-hour post-sizomycin treatment. Then when we compared this state with all the other states, so we found 45 genes that are differentially upregulated in cluster S4. And these genes are related to a pathway that implicate an immune-related genes. And here in the in situ hybridization, you can see, for example, the expression of I5-6 and IFID5 in the supporting cell layer 48 hours post-sizomycin treatment, while these 2 genes are not expressed in the control. So these genes have been identified and characterized during the second day post-sizomycin treatment. And if you are interested in this, so please check the publication that the paper that we published last year. So then we looked at cluster S3, and we found that cluster S3 contained mainly cells from the early time point, and especially 12 and 16-hour post-sizomycin treatment. So we concluded that cluster S3 contained the early responding supporting cells. So then when we compared cluster S3 with all the other clusters, we found an interesting set of 85 genes that are differentially upregulated in S3 comparing to the others. Then when we did a gene by gene curation, we found an interesting set of genes that are implicated in the mesequipal to epithelium transition genes. We found also 2 growth factors, 3 protein coupled receptor, and 5 transcription factors. Sorry. Okay, so and ingenuity pathway analysis revealed several signaling pathways and also an ERK5 signaling. So, and ERK5 signaling is very interesting for us because it's known to be activated by growth factors, but also has an important role in regulation of cell proliferation and cell differentiation. So we decided to look at this ERK5 signaling but also the expression of F2R1, which is a G-protein capsid receptor, and HBEGF, which is a growth factor. But we were also very interested in looking also at the activation of the receptor of HBEGF, which is EGFR. So we think that maybe the activation of EGFR can be an essential component in a signaling pathway that leads to proliferative hair cell regeneration. And to confirm our hypothesis, we decided to use a pharmacological strategy to screen the most prominent signaling proteins that are upregulated in responding supporting cells. And we decided to use S-phase entry as a quantitative readout. So the molecule that we decided to test were infused into the semicircular canal of P7 2 hours prior to sizomycin infusion. Then EDU was injected at subcutaneously at 24 hours and 42 hours for the assessment of the 48-hour time point. So we first confirmed that sizomycin induces a robust S-phase entry in the supporting cells, as you can see here. So we have here supporting cells that are SOX2 and EDU positive. And we have also the quantification that's shown here. And this is a confirmatory result. Then what we found is that when we infuse F2R1 antagonist in association with sizomycin, we found a significant reduction of EDU-labeled nuclei at 48 hours post-sizomycin and F2R1 antagonist infusion. So you can see this here, but also in the quantification. So we observed the same result using MMP inhibitor. Then using EGFR inhibitor, again, the same way. So we have a significant decrease in EDU incorporation in the supporting cells with ARC1 and 2, and finally with a STAT3 inhibitor. So what we concluded at this step is that F2R1, MMP, EGFR, ARC1 and 2, and STAT3 are signaling components that are essential for proliferative hair cell regeneration in vivo. But this posed the question of how are the individual signaling components are ordered along a potential signaling cascade. So for that, we decided to use QRT-PCR and to determine the dependencies. Sorry, I really don't know what's that, but yeah. So to determine the dependencies of gene upregulation for individual signaling events. And here we decided to focus on the expression of F2R1, on HBEGF, on MMP2, and also on GISP16. And I will go through each result independently. So first, what we found is that the expression of F2R1 mRNA expression was decreased by F2R1 antagonist itself, but also with MMP inhibitor, EGFR inhibitor, and ARC1 and 2 inhibitor, but also with STAT3 inhibitor. So this could be the evidence of a positive feedback loop that keeps supporting cells active and efficient. Next, what we found is that the expression of MMP2 and HBEGF mRNA was decreased by MMP, EGFR, and ARC1 and 2 in and EGFR inhibitor, but not with ARC1 and 2 and STAT3 inhibitors. So based on this result, we concluded that MMP2 and HBEGF upregulation depend on MMP and F2R1, but is independent of ARC1 and 2 and STAT3 signaling. So then based on all this observation, so what we think is that during hair cell demise, so when the hair cells are dying, and also we have this proteolytic activation of F2R1, so maybe HBEGF is an early component that is essential for hair cell regenerative process. And then with the presence of MMPs, but especially here MMP2, so HBEGF get released. So this is a shedding of HBEGF that go and activate its receptor EGFR. And our observation that MMP2 mRNA and HBEGF mRNA expression is not affected by ARC1 and 2 and STAT3 inhibitor means that maybe they can come as a downstream of this signaling that we are proposing. So, wow, I'm really sorry this wasn't like this. Okay, so yeah, but we are lucky we have the quantification here. I'm really sorry for that. So, okay, what we found is that sizomycin induces an increase in our phosphorylation of a 10-fold change of phospho-ARC1 and 2, and this is 24 hours post-zomycin treatment. Then when we use MMP, EGFR, but also F2R1, we observe a decrease in the activity of ARC1 and 2, of phosphorylation of ARC1 and 2, but not with a STAT3 inhibitor. So, and this means that F2R1, MMP, and EGFR, they come upstream of ARC activity, and STAT3 comes even downstream of ARC or is completely independent of the signaling pathway that we are proposing. So, and another gene that we found to be differentially upregulated in responding supporting cells is a MALP kinase phosphatase named GISP16. So what we found for GISP16 expression is that the mRNA of GISP16 is not affected by F2R1 antagonist and is not also affected by EGFR inhibitor. What we found also is that GISP16 mRNA is slightly increased using MMP inhibitor, but also using ARC1 and 2 inhibitor. And it's also slightly decreased, but not abolished, using STAT3 inhibitor. So what we concluded is that MMP and ARC1 and 2 activities tune GISP16 upregulation, and also that GISP16 might have a role in a proliferative response duration. So it plays like a balance here for in the ARC kinase activity. Okay, so here, so far what I showed is that F2R1, MMP, EGFR, MALP kinase, and STAT3 signaling is essential for supporting cell S-phase entry. But the other question was, what is the connection between inhibiting one of these components and the transcription factor candidates that we know that are differentially expressed in responding supporting cells? So by the same way, we used QRT-PCR, and what we found is that the expression of TCF24 and KLF6 mRNA was not affected by these different molecules that we have tested. And this led us to think that maybe TCF24 and KLF6 are implicated in another permissive pathway that we need to investigate. However, what we found is that ATF3, FOSL2, and CREAM are mRNA expression is decreased when we use an F2R1 antagonist, MMP, EGFR, ERK, or STAT inhibitors. And we concluded that these three transcription factors are dependent to the signaling pathway that we are proposing here. So as a conclusion for this part, we have shown today that supporting cell exhibits significant gene expression changes at 12 hours post-zomycin treatment when they are in proximity to dying hair cells, and that the F2R1, MMPs, EGFR, MALP kinase, and STAT3 pathway plays a pivotal role in activating supporting cells. What we found also is that there is two groups of transcription factors, one that is dependent to the signaling pathway that comprises ATF3, FOSL2, and CREAM, and the other one is completely independent. And here we are talking about KLF6 and TCF24. So now that we have identified the signaling pathway, the other question that we need to address is how this pathway is activated. So in other words, what is the communication between dying hair cells and responding supporting cells? And for that, I'm planning to use our single cell RNA sequencing and also use algorithms like single cell communication or cell chat. So to identify the pairs of ligand receptor and to identify signaling pathway network. So, and I believe this will give us more knowledge of intracellular communication network, but also identify some potential therapeutic targets for hearing restoration. The other open question that we have in the lab is what are the other permissive pathways? So as I mentioned, we know that we have two transcription factors, but we don't know why they are expressed. And this is the work of Austin, who is a newly graduate student in the lab. The other question is how do regenerated hair cells become reinnervated? And this is mainly the work of Mitsuo, who is a postdoc in the lab, and please stop by the posters this afternoon. So the other thing that we are working on right now is to translate the sizomycin-induced hair cell loss paradigm to the mouse cochlea. And for that, we have established an in vivo model that is equivalent to the Avian in vivo model. And this consists in unique injection of sizomycin into the semicircular canal of 1 month FEB mice. So this work has been in collaboration with Fabian and Aisha. And what we found is that after a single injection of sizomycin, we can induce a total outer hair cell loss within 24 hours, while the number of supporting cells is not affected. And we believe that this model can be used for further research in our field. But for us, so the aim of developing this model is to characterize or to evaluate supporting cells transcriptomically and epigenetically during hair cell, while the hair cells are undergoing the apoptotic process, but also at a chronic hair cell loss. And for that, we are planning to use the FBXO2 mice, where average cells from the cochlea expressed a venous expression. And we believe that this will help us to identify the highly abundant mRNA and active gene regulatory networks in supporting cells, which could reveal potential targets for a therapeutic intervention. And with that, I think I was very quick. So with that, I would like to acknowledge you all, thank you all for your attention. And I would like to thank the Heller Lab, my mentor, Stefan Heller, who is here, all my colleagues, the Gen Lab to be very great collaborators, also the OHNS microscopy facility, the functional genomics, all my fundings, the lab's fundings, and also the Bellucci Foundation, and again, the organizers for giving me this great opportunity to be here today. Yeah, and I will be very happy to take any questions.
So in this system that you find out so well described in the chicken, what about those genes in the mammalian cochlea and in the mammalian vestibule? Is there any, have you looked into why are they or are they not, or, you know?
So what we were very interesting in looking at was the F2R1, and I believe that F2R1 is expressed at very low level. For the utricle, I need to check. I did not. I don't know in the utricle if this gene is expressed. What are for the transcription factors? Yes, they are expressed as well. Maybe not in the during. So we don't know what happens when the hair cells are dying, especially in the supporting cells. So it's something that we need to investigate. And yeah. So, but I really don't know.
I mean, obviously, you could try to reconstitute the pathway.
Yes.
In a mammalian system, but that would be maybe very hard. But
we need to try.
Basant question comes from the online session. Did you test ABR and DP after sizomycin treatment of the chicken and mouse models?
Yes. So Mitsu helped me to do that. So we tested ABRs and DPOE after sizomycin. So of course we observed like complete hearing loss, and then we tested also with different drugs, and we saw that it doesn't interfere with hearing, so they have perfect hearing function. Yeah. Okay. Thank you so much.